So this is histology part one. So let's talk about a little bit about what histology is before we get too far. So histology is actually the study of tissues. Histio means tissues, and that's what we're going to look at. So if you look at this slide over here, this is the slide of the esophagus. And so the esophagus is an organ. And if you remember our pyramid, we talked about it yesterday. If you look at the pyramid, remember we start down here at the chemical level. Well, there's a chapter for chemistry in our book, but we're going to skip it. Now, hopefully you've had chemistry. If you have not, you might want to read this chapter, but that's chapter 2. And then the next chapter in the book, and if you move up on our pyramid, it's the cellular level, so cells. And this is chapter 3 in the book, and we're also going to skip that as well. So you spent a whole lot of time in general biology talking about cells, and so we're not going to cover that. It doesn't mean that you're not shouldn't understand it and remember it. You should. But the next level up is the tissue level, and that's what we're going to be starting to talk about today. And that is chapter 4 in the textbook. If you go up one more, that's the organ level. And so if you remember, the organ is made up of a bunch of different tissues. Well, the esophagus is an organ. And so when you look over here, you'll see a bunch of different tissues. This tissue is different from this tissue, which is different from this tissue, which is different from these tissues, and so on. And so right now when you look at it, you may just see a bunch of purple stuff. But by the time we finish talking about tissues, you should be able to figure out what these are. You should know that what this tissue is and where you would find it and what its function is. The same thing for these tissues and so on. Any questions about that so far? Okay, so what is a tissue? Well, basically a tissue is just a bunch of cells, but it's a bunch of cells that have some commonalities. In other words, they're similar. They have a similar structure, they have a similar function, and they come basically from the same place. They have a similar origin. Well, there are only 4 types of tissues in the adult, just 4. There's epithelial tissue, there's connective tissue, there's muscle, and then there's nervous or nerve tissue. Well, today we're going to talk about epithelial and we're going to talk about muscle and nerve. We're going to save this one for another day because this one is has the most tissues in it. In other words, it takes the most time, and so we'll devote a different day to it. But these are the ones we're going to cover today. And the first one we're going to look at is epithelial tissue. So first of all, epi, that prefix, if you ever see it with anything, what it means is on top of. So this tissue is on top. It's on the top of something. And so if you look at a bunch of tissues, this one right here, the one that's on the top, is going to be the epithelial tissue. It's a surface tissue. It's never deep. It's a surface tissue. And so when we go to look at it, we're going to find it all over the whole body, but it's going to be the boundary. So it's the boundary between different environments. So it's right here. So here's the outside and here's the inside. It's going to be right there. But it's also inside here because that's a boundary. It's inside here. It's inside here. It's always on a surface. It's always on a surface. It's always on the top. And so it's always going to face a space. And so we're going to see that, and that's how we're going to find it. And so some examples of this tissue are things like the skin, and then the lining of GI tract and other organs, the lining of blood vessels, the lining of the heart, all sorts of things. It has a bunch of different functions. It can protect, but it can also do things like secretion and absorption. It can also do things like filtration. And so those are the kinds of things that we're going to see. Well, these are very different functions. And so to be so different functions, we're going to have different types of epithelial tissue. And there are different types. And we're going to look at them and we're going to figure out how to name them. So what we're going to learn and know about tissues, and not just epithelial tissues, I want you to be able to recognize them. In other words, if you see a picture or a diagram or a photomicrograph, you should be able to recognize the tissue. You should be able to recognize it from a description. So you should be able to use words to describe what the tissue looks like. And then we also want to know what it does and where it's located. So we also need to know these. These are the characteristics about it that are important. And so we're going to do that with every tissue. But again, we're going to start with epithelial tissue. So let's look at epithelia. Well, we want to classify them or name them. Well, epithelia are named pretty easily. There are two criteria that we're going to use to name an epithelial tissue. And the first one is we're going to look at it and we're going to ask us how many layers of cells there are. How many layers? And so there's either going to be one layer like this, or there's going to be more than one layer like this. Well, if it's one layer, it's called a simple epithelium. If it's more than one layer, it's called a stratified epithelium. Stratified literally means layers. So it has more than one layer. And then there's a special one. We'll get to it in a little while, but look at it. It's called pseudo-stratified. Well, pseudo means false. So in other words, this tissue is false stratified, false more than one layer. When we look at it, you'll understand what we're talking about, but it's false. It's not stratified. It's a simple epithelium because it's not more than one layer. It's false. But let's look at these. So here's a simple epithelium. So look at it. It's one layer. Here's a stratified epithelium, more than one layer. And then here's our pseudo-stratified. Well, when we talk about layers, we're going to have to figure out how to see the layer. And so in order to do that, we're going to look for two things. One thing we're going to look for is a basement membrane. And all epithelial tissues have a basement membrane. This basement or basal membrane is not alive. It's really like a layer of glue. And it holds the tissue onto the other tissues below it. So here is this basement or basal membrane. Again, there are no cells in here. It's not living. It's like glue. And then here it is right here. You can see it and so on. And then the other thing we're going to look for is an empty space. There's space out here. There's space right here. And there's space here. That space could be the external environment. It could be the inside of a cavity. It could be all kinds of things, but it's space. It's empty space. Or it could have some sort of component in it, but it's space. And so the top of the cell that's facing the space, the word for top is apex. So this is called the apical surface. And here, there's our apical surface, the top. And here's our apical surface, the top. So what we're going to do is we're going to find those two things. We're going to find the basement membrane, that's it right here, and we're going to find our apical surface right there. Here's our basement membrane, here's our apical surface. Here's our basement membrane, and here's our apical surface. And then what we're going to do is we're going to count how many rows of cells there are between those. So look, between here and here, there's one row of cells. There's the row right there. That's one cell. They're in a row. But if I look at this one, I count 1, 2, 3, 4, 5, which is more than one. If it's more than one, it's stratified. And then if I look at this one, it might look like there's more than one, because what people tend to do when they count a layer is they tend to line up nuclei and call that a layer. Well, that works fine for all of these. But look at this. The nuclei are in different levels in the cell. And so if I line up nuclei, it looks like there's 3 layers there, or maybe even more. But there's not. And the reason there's not is because every single cell touches the basement membrane. Look, this cell comes down here to the basement membrane. This one comes down here to the basement membrane. This one, even though only a little bit of it makes to the top, it comes down here to the basement membrane. And that way is all of them. So what confuses us or tricks us is that the nucleus is at different levels in the cell. So the level varies. And if you line up nuclei, it's going to confuse you. So it's fake, stratified. Does everybody understand those? Can everybody see the simple, stratified, and pseudo-stratified and why they're called that? So here's another view. So look, there's our basement membrane right there. And then here is our apical surface right here. Well, look, it's one row. So that's a simple epithelium. But look, this is also, there's the basement membrane, and there's our apical surface. This is also one row. So it's a simple epithelium. There's the basement membrane. There's our apical surface. It is also one row, even though the nuclei are at different levels. So all of these are simple epithelia. And then we're going to ask ourselves a second question. And the second question is, what are the cells shaped like? And so when we look at it, we're going to see 3 different shapes. We're going to see a shape that's flat. They're going to look a little bit like fried eggs. If you've ever seen a fried egg, if you know what it looks like, from the top, it looks like this sort of, and there's the yolk. But if you look at it from the side, it looks very, very flat with the yolk bumped up a little bit. And that's exactly what these look like. If they look like that, they're called squamous. And then we have these, which are sort of boxy shaped or cube shaped. And they're not just flat. Remember, they're cubed. So they actually have 6 sides. They look like this. So if you look at them, they look like that. And they have a nucleus that's round, and it's usually right in the very center. These are cube shaped, so they're called cuboidal. And then we have these, which are tall and skinny. And tall and skinny is kind of like a column, so they're called columnar. They tend to have oval nuclei, not round, but oval. And the nucleus is closer to the bottom. So here's squamous. Look at it. There's from the top, and there's from the side. There's cuboidal. There's from the top. There's from the side. And there's columnar. There's the top, and there's the side. So again, this looks like a fried egg. This looks like a cube. And this looks like a column. And then look, there's our round nucleus, and it's right in the middle. And here's our oval nucleus, and it tends to be down at the bottom. Can everybody pick out the different shapes? So to name these, all we're going to do is combine those 2 things. And so if we have one layer, remember it's simple. If we have more than one layer, it's stratified. And so we're going to combine them. And so when we do that, we're going to get simple squamous, simple cuboidal, simple columnar, and then pseudostratified. And the only pseudostratified ones there are columnar. So these are the simple epithelia. Now, a little bit later, we're going to do the same thing for the stratified ones. But let's look at these first. So let's look at simple squamous. So without knowing any of this stuff here, we know a lot about it. It's simple, so it's one row. It's squamous, so the cells are flat. You should be able to picture that in your head. One row of flat cells. And that's exactly what we see here. That's the description of it. Well, what these are good at doing is diffusion. Diffusion, because remember, diffusion takes place across short distances. So here are these cells. They're not very big. And so it's easy for materials to move across these cells because the distance is short. So they're good at doing diffusion. They're also good at doing things like osmosis and filtration. But the word you need to remember when you talk about their function is diffusion. And then, not only that, these cells provide sort of a very slicky surface, which reduces friction. And so things slide along here really easily. It just slides like a kid sliding down a slide. And some of the things that slide across it are things like blood. So blood cells just slide across it. And then here are the places where you find it. So let's look at it. So this is found in your lab book. And so if you look in your lab book, and we'll come back to lab later on today, at exercise 6, what you're going to see is there's going to be a page like this for every single tissue. And look, it's like a box. And inside that box is basically everything you need to know about that tissue. And so it's a great place to study tissues if you use your lab book. But look what's in here. First off, there's a picture, and so we can see what it looks like. There's a description, so there's our description. There's a diagram of it, so we get an even better view. And then here are the functions and the locations. So basically everything you need to know about this tissue is in this box. But let's look at it. Let's look over here at the picture. So what this is in the lungs, and the lungs have little tiny sacs in them like this. They're called alveoli. And what we're looking at are alveoli that have been cut. So there's one alveolus. It's a sac. It's not a cell. Look, these are the cells right here. That's a cell. And there's its nucleus. Same thing right there. That's the cell. And there's its nucleus. And so if you look at these cells, you can see just how thin and wide and flat they are. And so they're very, very, very thin. And so if you drew one from the side, it's like this, very thin. And then it's got that little disc-shaped nucleus. So again, they're going to be really good at doing diffusion. And what happens when along with diffusion is oxygen diffuses in one direction and carbon dioxide diffuses in the other direction. So it's going to diffuse across these membranes. And if you look over here, you can see the picture, or the diagram rather, and you can see how easy it would be for materials to move across. Now in this photo, it's really hard to find the basement membrane. You can see it over here in the diagram, but because this tissue is so thin, the basement membrane is very thin and also hard to see. But if we blow it up, it looks like this. So here's a picture again. There's our basement membrane. Here's our apical surface. One row, flat cells, so it's simple squamous epithelium. And look, if we look at this photograph, it's not this down here. It's not that. It's just this right here. Remember, it's on the surface. So we're going to find these two things. We're going to find the apical surface here. Here's that space. We can see it. And even though you can't see it very well, there's a basement membrane right there. And in a different picture, you would be able to see the basement membrane much better. Remember, if you look at it from the top, it looks like this. They look like fried eggs. But from the side, they look like this. So that's simple squamous. So look at this picture, and you'll see a bunch of different tissues, but the only one we're interested in is this ring right here, this. Just that right there. That's the simple squamous. And it goes all the way around. Look, it makes a circle. It's actually a cup-like structure, and it's found in the kidney. Now, if you take AMP2, you'll talk about it. That's Bowman's capsule, but it's not this stuff out here. Not any of this. We have to find an apical surface. There's our space right there, and right here's the cells. Can everybody see it? Can everybody figure it out? Okay, let's look at simple cuboidal. So already you should know something about it. Simple, one row. And cuboidal, cube-shaped cells. So you probably could draw this. It's just a single row of cube-like shells. Remember, they have very large spherical nuclei, so they're located right in the middle. So they look like this. That's what the cell is going to look like. Well, they're too big or too tall for diffusion to happen, because diffusion only happens across short distances. And so they're not good at doing diffusion and things like filtration and osmosis. What they're good at is active transport. They do secretion and they do absorption. So these cells actually have carriers that move things across by active transport. And remember, active transport requires ATP. So you're going to find them in places where that's happening. So you find them in places like the kidney tubules, glands, things like that. And when we look at them, this is what they look like. So look, single row, cube-shaped cell, central nucleus that's round, it's spherical. And then if we look at them for real, again, here's our box. So there's our photomicrograph, there's our diagram, there's our description, there's location and function. Again, we need to find the basement membrane, that purple thing right there. Find the apical surface. We're going to do that on the the slide as well. Well, look at this. It's sort of a flat surface. The cells are all like this flat sheet like this. But think about what would happen if I took this flat sheet and hooked the ends together. It would become something that looks like this, and it would be hollow in the middle. And the cells would be out here like this. So in other words, it would become a tube or a tubule. And that's exactly what we're seeing in this picture here. So here's a tube, and it's hollow in the middle. Here's a tube, hollow in the middle. You can see it over and over. And these purple lines that you see, That's the basement membrane right there, that basement membrane. And then up here, this space, that's where the apical surface is. So if you look at these cells, there's a really nice one. That's a cube-shaped cell. It's just one row. If we blow it up, it looks like this. So you can kind of ignore this stuff out here for right now. But look, there's our basement membrane right there, the little purple line. There's our apical surface right there. And look, what we have is a tube or a tubule. And these cells, there's the cell right there, cube-shaped, big round nucleus. Cube-shaped, big round nucleus. And so you can see that here, you can see it over here, and you can see it over here. And look how perfect these are. That's a cube, a cube. There's another picture. But look, now the tube has been cut long ways. Remember what we said that's called? It's a longitudinal section. But still, look, there's the cube. There's the basement membrane, that purple line. Here's our apical surface right there. So one row, cube-shaped cells. If we blow that up even more, it looks like this. So here's the space. Here's the space. There's the basement membrane, and there's the basement membrane. Well, when you look at this, you might be tempted to think that this is more than one row, but it's not. Because there's the basement membrane, and there's an apical surface on each side. Really all this is two tubes laying next to each other. Here's one tube, and here's one tube. That makes sense to everyone? Okay, let's look at simple columnar. So again, without knowing anything about it, we can figure it out. It's one row, tall cells, oval-shaped nuclei down at the bottom. So it's going to look like this. Now they're way too tall for diffusion and osmosis. So again, there's no way that can happen. So these are also good at doing secretion and absorption. And again, it's active transport. And so they're going to use ATP. Columnar cells often have another kind of cell with them, and these are called goblet cells. So goblet cells are actually tiny little glands that secrete mucus. So if we look at it, we're going to see the columnar cells, they look like this. Here's our nuclei down at the bottom. And then every so often, we're going to see a cell that looks like this. And this cell is going to have bubble-like things in it. Well, what those are vesicles containing mucus, because this secretes mucus. And the mucus then covers the surface of the whole tissue. So when we look at it, we're going to see something that looks like that. So here's our basement membrane, here's our apical surface. These are the columnar cells, one row, and then there's a goblet cell. And they're supposed to look like cups or goblets, and they kind of do. And then here are all these little vesicles with the mucus in it. And so the mucus is going to come out, and it's going to cover the surface here. And so if we look at a picture of that, here's our box. So there's our photomicrograph and our description, location, function, all of that. And here's the tissue. We have a very well-developed basement membrane here. Really easy to see. And then there's our space right there. So here's our apical surface. And look at this. These are tall, skinny cells. And the nuclei are all in a row. They're not at different levels. They're all in a row. Now, this one's been cut away, but it would be there. You can see them over here. So, simple columnar. Same thing here. Here's our basement membrane. Here's our apical surface. These tall, skinny cells with these oval-shaped nuclei. Any questions about simple columnar? Okay, let's look at pseudo-stratified columnar. So remember, pseudo means false, stratified more than one layer. And so even though it's a single layer of cells, it makes it look like there's more than one because they have different heights and the nuclei are at different levels. And so this is columnar tissue, and remember columnar often has goblets with it, and we're going to see goblets here as well as with that other one. The tops of these cells can be ciliated or not ciliated, but the only one I'm going to show you is ciliated. So a trick to knowing pseudo-stratified, at least in this class, is that they're ciliated, and they're the only tissue that is ciliated. So if you see cilia, it's pseudo-stratified. So if you look at this, you can see cilia right there, cilia. And remember what cilia are. They're little organelles that can move. They produce movement. Well, it doesn't move the cell like in a paramecium or something. What it does is it moves mucus. Because look at all these goblet cells. There's a goblet cell right there, and it's going to make mucus, and the mucus then is going to get moved by these cilia. If you look over here, you can see why it's pseudo-stratified. The nuclei are at different levels. And if you look at this picture, if I were to just count layers, I'd probably say that there's at least 4, maybe 5 layers in here. But there's not, because every cell touches the basement membrane. They may not always go all the way to the surface, but they all touch the basement membrane. So here's a diagram of it. So you can see these cells. So again, if I were to just look at it, I'd say there's at least 2 layers, but there's not, because every cell touches the basement membrane. But the easiest way to identify this tissue is to look for cilia. And again, cilia are going to move. And then here's our goblet cells. And remember, goblet cells are going to make mucus, so the mucus is going to come out, and then the cilia is going to move the mucus. These are mostly found in the respiratory tract and in the reproductive tract. So that's where they're mostly located, in the respiratory tract and in the reproductive tract. So everybody see the basement membrane? Everybody see the apical surface? Now look, I would count 1, 2, 3, 4 lobes, but they're not. And we know that, at least in this class, because what are these things? Anybody remember? Cilia, that's right. And this is the only tissue we're going to see that has cilia. So, pseudo-stratified columnar. There's another view of it. This one you can see the cilia really well. They look like hairs, but don't call them hairs. They're not hairs. You can say they're hair-like, but they're not hairs. Okay, so those were the simple epithelia. Any questions about the simple ones? So we had simple squamous, simple cuboidal, simple columnar, and pseudo-stratified columnar. Any questions? Okay, well, let's look at the stratified columnar. So remember what stratified means. All it means is there's more than one layer between the basement membrane and the apical surface, more than one layer. But remember, not only do we count layers, we're going to look at the shapes of the cells as well. But there's a problem with stratified epithelia because the cells at the bottom don't necessarily look like the ones at the top. So if I look at this, these at the bottom look cuboidal, but when I get up here, they look different. So we're going to have to decide where to look to determine the shape, because we're going to get a different name if we look here versus here. And we don't want different people using different names. And so what we use is the top. So we're going to look at the top layers to determine the shape. So the top layers of this one are squamous. So that would be stratified squamous. We're always going to look at the top, not the bottom. And then we're going to come up with our different types. And so we have stratified squamous, stratified cuboidal, and if you were in the lab, they're not set up. Straticolumnar, also not set up. And then transitional, which is another kind of weird one. So let's look at stratified squamous. So without knowing anything about it except its name, we already know that it's more than one layer. In fact, it's lots of layers. And we know that the top cells, not anywhere else, but the top cells are flat. Well, because it has so many layers, it's thick. It's thick tissue. And so if I damage the outer layers, I rub on them or break them, damage them or whatever, they may actually come off. But I still have this much tissue before I get down into the rest of my body. And so the function of all stratified tissues is protection. It protects the underlying tissues, the underlying areas. And you find it in places where abrasion happens, rubbing, scratching, some sort of abrasion. Well, when we look at stratified squamous, there's two subtypes. So they're both stratified squamous, but they're different from each other. One of them is called wet, and one of them is called dry. My mouse is dying. Maybe it'll last through the end of the lecture. But they're called wet and they're called dry. What makes them wet or dry is something called keratin. So keratin is a protein. And keratin is what our fingernails are made out of. It's what our hair is made out of. It's also in the outer layers of the skin. And keratin is a little bit waterproof. So if you put a layer of keratin here, the fluid in your body cannot make it to the surface. It stays on the inside. And so if I touch the outer part, there's no water on it, so it feels dry. So everybody touch your skin. It's dry. And that's because of this, because of keratin. But there are other places where we have abrasion that we don't have keratin. And those, there's no layer to keep the fluid out. There's no layer of keratin, so the fluid, the water of your body, makes it all the way to the surface, and it collects on the surface like little drops. And so if I were to touch that, it feels wet. So this is like the inside of your mouth, or the inside of the esophagus, or the anal canal, or the vagina. So two different types. We have keratinized, which is dry, and we have non-keratinized, which is wet. They both have lots of layers, and they both protect against abrasion, but they're located in different places. So let's look at the two types, and let's look at the non-keratinized first. So remember, this is the wet version. So you find it in places that are moist, that are wet. You find it in the mouth, the esophagus, the vagina, those places. What's going to happen is lots of layers of cells, and the cells are going to move up as new cells are produced, and eventually the cells are going to slough off. And so when we look at it, we're going to see a bunch of cells. So down here is our basement membrane. Up here is our apical surface. And the cells move this way. And then eventually they're going to slough off. They're going to come off. Well, the way to tell that this is non-keratinized versus keratinized is we're going to look for nuclei in the outer rows. If nuclei are present, it's non-keratinized. So look, down here we have nuclei, but that's not where we're going to look. We're going to look here. So look, there are nuclei in the outer rows. So this is non-keratinized. But look here. There are nuclei in the outer rows, non-keratinized. Also look at this. If we look at the cell shapes, we look down here, look, these don't look squamous. They look cuboidal or something, but we're going to ignore those. We're going to look at the shapes here. That's where we're going to look for the shape. And if you look at the shapes of these, they're thin and skinny and flat-like. They're squamous. And then not only are we going to look at the shapes here, we're going to look for nuclei here. And so there are nuclei present. So this is stratified squamous, non-keratinized. And if we look at this, look at all the nuclei here. These cells are flat. You can see them over here pretty well. This is stratified squamous, non-keratinized. And look how thick this is. So you can see how it would be protective. It's thick. It's like wearing a thick coat versus a thin shirt. It's protective. Let's look at the keratinized version. So remember, there's this row of keratin that the water can't get past. And so the cells that are up here above the row of keratin are going to starve to death. In other words, they're all going to be dead. And when they die, their nuclei are going to disintegrate. And so there are going to be no nuclei, none, in the outer rows. No nuclei present. The outer rows are dead. And so when we look at it, it looks like this. So look, you can see here, There's where the basement membrane is, not here. That's where the keratin starts. So look down here and we can see nuclei. But if we look up here in this, no nuclei at all, not one. These rows are dead, and they are also going to slough off. But this tissue, which runs from here down to here, is stratified squamous, keratinized. Here's another picture of it. If you look here, you can see no nuclei. No nuclei. So if we compare non-keratinized and keratinized, look over here, there's our nuclei in the outer rows. But here, not one nucleus, no nuclei. This tissue runs from here down to here. There's the basement membrane. This tissue runs from here down to here. There's the basement membrane. If we blow that up, look, no nuclei, none. So, what do you think this is? Is it keratinized or non-keratinized? Here's the tissue from here to here. What do you think it is? Anybody want to guess? You can see the sloughing off right here. That's where the sloughing off happens. Yeah, Edenia says keratinized, and that's exactly right. And you can even tell exactly where the keratin starts. It starts right here. That's where it starts. Look, there's nuclei down here, but not one nucleus above it. Okay, let's look at stratified cuboidal. So again, without knowing anything about it, we can already figure out something. It's more than one layer, and the cells are cube-shaped in the outer rows. The reason we don't set this up in the lab is because it's a rare tissue. Now that doesn't mean only a few people have it. Everybody's got it. We just don't have much of it. It's only found in some glands. And typically it's only 2, maybe 3 cell layers thick. So if you look at it, it looks sort of like this. So look, these are cube-shaped cells up at the top. Very few layers, but there's more than one. So stratified cuboidal. And if you look at what it really looks like this. So there's the layers right there. Very few layers. Let's look at stratified columnar. It's also one of these rare tissues. And so stratified columnar, more than one layer. The top cells are going to be column shaped. They're going to be shaped like this. This is usually 3 or 4 rows of cells. So when you look at it, there it is, maybe 5 or 6. But anyway, if we look down here, it does not look columnar, but look, these look columnar. We're going to look at the top. And if you want to see what it looks like for real, it looks like this. So there's the tissue right there. And again, down here, these don't look columnar, but these do. And then we have one more tissue, and this is called transitional. The reason it's called transitional is because it changes. It changes. And what makes it change is stretch. So think of a balloon, like the kind you buy in the store. If you just take the balloon out of the package and it doesn't have any air in it, and you look at it, it looks dark, it's thick. But when you blow it up and you put air in it, the tissue stretches. And if I look at it now, I can almost see through it. That's how much it's stretched. And the more you stretch it, the thinner it gets. That's exactly what happens with transitional epithelium. It's only found in one place. It's only found in the urinary tract, mostly the urinary bladder, but also the ureters and part of the urethra. But it's in the urinary tract, and the urinary bladder is like a balloon, exactly like a balloon. But instead of putting air in it, what we put in it is urine. So it's more like a water balloon, but the more urine you put in there, the more the walls stretch. And as the walls stretch, the tissue gets thinner and thinner and thinner. And you can see it do that. So if you look here, this is what the tissue looks like in the unstretched state. So look at it. The tissue is fairly tall. There's the tissues, this tall. And the cells are tall and round and like this. And if you look up at the top, they have rounded tops, which is also different about this tissue. They're called dome cells. They have a dome on them. And this is the only tissue that has these domes. You can see them very easily here. Over here, you can see them very well as well. But if I start stretching this tissue, it's going to thin out. And we're going to go from this, here's the unstretched, to this, here's the stretched. And look, they don't look the same at all anymore. Look how tall this was. And now look how thin this is. Here we have these domes up here. Look, the domes disappear. They're not there anymore. So this is a confusing tissue because it changes. So to make it less confusing, the only way you're going to see it from me is like this. You're only going to see the unstretched. It's the easiest to identify. It's the one where you see the domes. Nothing else looks like that. Has these domes. So if we look at it here, there you can see the domes very well. And look how many layers it is. That many layers. It's a stratified tissue with domes up at the top. So you can't really confuse it with anything else because nothing else has these domes. See them here? The tissue runs from here to here. Any questions? So what I'd like to do is take a short break, and I'm going to recharge my mouse while we do that. So I'm going to stop the recording here, and then we'll come back. Let's take a break about 15 minutes, and we'll come back and do the other parts of it. Does anybody have any questions right now?